Hello, everybody. My name is Alex, and today we have a webinar. It'll be getting started soon, and it will be presented by our CEO, Vinit. And the webinar is being recorded, so it'll be available on demand in the future. And if you have any questions, they'll be answered at the end, and you can also put them in the chat.
Right. Good morning, everyone. Thanks for joining us today. And today we are going to be discussing this webinar is on the principles of radial flow chromatography. Some of you are familiar with it and may have questions, in which case, please ask. If there are any questions, please ask. Otherwise, I'm just going to go. The principles of radial flow columns, problems that they solve, applications of the radial flow columns, and we have embedded in the presentation, a couple of videos showing how the columns actually work. So right on this front page, you can see the animation of the radial flow column. Generally shows you how the flow is actually taking place in the columns. It doesn't go longitudinally through the column, but the columns are like a cartridge where the flow is applied all the way from the outside in. So before we get started on the subject of the columns, a little bit about Sepragen for some of you who are not familiar with the company. The company's been around for well over 2 decades, and we have a whole suite of products for downstream processing, well-established products for downstream processing. Certainly, we are inventors of radial flow chromatography, which we'll talk about today, but we also have a whole line of chromatography systems and skids going from the bench scale all the way to large scale that are used in GMP manufacturing. And they can be used with radial columns or any other columns. We also have single-use flow path chromatography systems, a lot of which are actually being used in gene therapy because we start with small flow rate sizes, which are bench scale, and scale up to large scales, which are flow-mounted skids with disposable flow paths. And lately, which will be the subject of forthcoming webinars, is a very exciting technology that we are introducing into the market, which is our QuantaSep continuous chromatography capture and flow-through systems. So here, we've taken the leadership, and we are providing means for people to do multi-column continuous capture, particularly for antibodies. And then we also have a flow-through skid. And then what we've also done is developed a buffer conditioning or buffer blending systems. And in terms of intensification, we have integrated these with continuous chromatography. So if people have interest in that, it's not the subject of today's webinar, but certainly reach out to us, and we'd be more than happy to set aside time to do a one-on-one and talk to you about some of these other technologies. So our products are already used to make commercially approved biologics. So it's not something that's used just in GMP or in the lab or for clinical production, but this is actually used, and the radial flow columns in particular are used to make 24 commercially approved drugs, biologics. So it's not a question of whether these things work or not. I think that whole question was answered 20 years ago. These things are well established, they work, they have benefits, and the choice for you is whether you want to use them to derive the benefits or not. And today there are over 200 global installations, and over $10 billion worth of biologics are used today made using these for commercial GMP production of biologics. Our company is based in Union City, California. And these things are US made. They're made here. These columns, all our equipment is made in the good old USA. What you see here, just the product range, what you see here on the left-hand side is a commercial installation we had a company that recently installed a $1.5 billion facility outside of Atlanta, and we were recognized by them as one of the key contributors or vendors to this operation. So these are not tanks. These are 4 large radial flow columns that are working together um, with total of, I think, 14 or 1,500 liters of column capacity. And on this installation, they're doing about 15,000 liters per batch. Okay. We also make chromatography systems, and this one particularly is a chromatography system with integrated buffer dilution, and we are running about 1,000 liters per hour on this machine. And then we have single-use purification systems, again for clinical and commercial manufacturing, where the entire flow path, along with the sensors and so forth, can be removed. Software that is being run is 21 CFR Part 11 compliant. And then these columns have also been used in the dairy industry, actually in the high value nutritional industry, to produce lactoferrin from milk or whey. Lactoferrin is being used for the next generation of infant formula, and lactoferrin gives the baby better mental acuity and also provides for other health benefits. So this is the latest range of infant formula that the large companies have come out with, and our columns are being used to process well over 100,000 liters of milk a day, and we just completed an installation of 15-ton annual capacity of lactoferrin. And we are trusted partners with large biopharma companies who have used our products in different stages of their operation. Some drugs have made it, others have not, but regardless, the technology has been widely used, and we partnered with various companies, so which means that we have in-house manufacturing, not only manufacturing ability, but all the documentation and all the traceability information and all that that's needed for GMP manufacturing. So, to the columns. Why the need for radial flow columns? Well, particularly in the biotech industry, as titers have gone up, slow flow rates, and as titers have gone up, it's necessitated the need for larger and larger columns downstream, and downstream processing has become a bottleneck. So slow flow rates in conventional columns have led to large cycle times, often greater resin usage, because obviously the columns have to get bigger columns, larger footprints, larger facilities. And with the increase in size of these columns, the conventional columns, you don't scale up or higher, you scale out or wider, and the additional facility, and it takes, it's very unwieldy to pack these large 2-meter columns. And so, and often there isn't space, or if the facility is on the second floor, the floor doesn't take the weight of these huge columns. So regardless, there are lots of issues, as I'm sure many of you are aware. And so the radial flow columns provide a convenient way of not only getting higher productivity, but reducing footprint, reducing waste, and also, in many cases, reducing resin usage. And ultimately, of course, reducing cost of purification. So it's a low-hanging fruit. You can try to do this by continuous chromatography systems, which we provide, or by changing resins or so forth, but it's so much easier just to change out the column. And these columns, people have made these changes on columns. Not only you don't have to develop it necessarily in PD, but these changes have been made in later stages of clinical trials or even post-approval. Not very difficult change. So the benefits of the radial flow columns are short cycle times, particularly with protein A, ion exchange, affinity, and HIC. The radial flow design is such that you're using short beds to go fast. I mean, nowadays, people are using ion exchange membranes, which are very, very thin beds. So we all know that short beds are fine. You don't need 20, 30, 40 centimeter beds in order to do a separation. particularly with binding, non-binding separations like ion exchange or affinity. Small footprint. You don't need large footprints. These columns are very small footprints. And typically, per square meter of area, you can get 4 to 10 times higher flow rates. So without having to change out your resins, change out your process, very simply by changing out just the column, you can get, orders of magnitude. You're not talking about 10, 20% increase in productivity, but an order of magnitude or multiple increase in productivity. Operating pressures are low, which is very useful because pressure drops are a bane in large-scale processing. And the other thing is that the way you pack or run small columns is exactly what you do on a large column. And we use a slurry packing technique. We don't pour resin into the column and move adjusters up and down. It's very hard to do that on large 2-meter columns. Standing there with buckets and pouring resin into the column and moving adjusters and using hydraulics. Here we just simply slurry pack the column. And the little lab column and the large column is packed exactly the same way. So it's very scalable. And in many cases, we get better performance, lower pool volumes. And the scale up is linear, truly linear, which means that from a 100 mil column, if you go to a 100-liter column, you multiply everything by 1,000. You multiply the loading amount by 1,000. You multiply the flow rate by the back pressure, and the separation time is exactly the same. And of course, all that means you reduce time to market, you reduce your experimental errors upon scale-up work you have to do, and ultimately you reduce the cost of purification. So we'll show you a little video. This is one that a distributor in Europe made. I'll share this with you. I think it illustrates very well the principle of the radial flow column.
Welcome to our series of tutorial videos from INL Biosystems. Today we will present the principles and benefits of Sepragen's Superflo radial flow column. To explore the key benefits of this technology, we need to observe the fundamental differences in design and flow physics of the radial flow column. Unlike a traditional axial column, the sample is applied along the outer wall between the outer column frit and column body. The outer frit contains 1 or 2 porous concentric cylinders. The eluent and the sample will enter the chromatographic bed through the outer frit and travel through the column horizontally. It is then collected inside the inner frit and eluted through the bottom outlet. Using the Superflo radial flow technology, the bed height is calculated as the distance between the outer and inner frit. Superflo's radial flow column drastically increases your productivity, helping you reduce your bottlenecks in biochromatography. The radial flow's unique design offers a significantly larger sample inlet area. Even at higher overall flow rates, the column's radial design results in lower linear velocities, which increase the interactions between the sample and the resin, resulting in higher quality separations without compromise. An additional intrinsic design of the Superflo column is the simplicity of linear upscaling from the lab to production scale. As the system pressure does not increase with the column size, this eliminates the complexities of linear upscaling, resulting in data from the small scale that is more representative of what can be achieved at the larger scale. Wouldn't it be nice to accurately predict what a 350-litre column behaves like before spending €250,000? Our wedge column represents a cut-out version of a much larger column to explore processes before scaling to production size. The Superflo columns are available from 50 millilitres both in acrylic and stainless steel up to 500 litre industrial production ranges, with the flexibility and engineering services to accommodate custom builds and special projects. Superflo represents a superior technology for applications such as affinity and ion exchange chromatography. This is complemented with the simplest packing protocol of any column commercially available on the market.
So this just gave you a quick glimpse of the radial flow columns and our upscaling and our scale up from the bench to the large scale. And this diagram basically represents how the flow goes through the column. So it comes in through the column inlet and is diverted to the entire outside wall of the column, goes through the outer fret, through the resin bed, through the inner fret, cascades down the side of this inner concentric cylinder, which is inside the inner fret, then comes out the bottom. So you have a very large area and a very short, which is the outside fret area, 2 pi r/h, and the distance between the inner and the outer fret represents the bed depth. Okay? And that's how the flow takes place in the column. So basically, if you look at these two cylinders, they're the same volume, but the outside area is much more, in this particular case, on the top, which is the radial column, than the 2 pi r/h, as opposed to the pi r squared in the axial column. And then with conventional columns, you take a narrow column in which you do your chromatography, but when you scale up, you don't actually have, even though you might keep the same bed height, the ratio of the diameter of that column to the bed height is not constant. So it's not a good model for scaling up these separations, because as you make the columns wider, what happens in a conventional column, if you go to a 2-meter column with a 10-centimeter bed depth, then you have to go 1 meter in this direction, 10 centimeters down, and 1 meter back. Whereas part of the flow is just going down y, which is 10 centimeters. So now it's obviously the pressure drop in this circuit is more than the pressure drop down the center. And so you experience a parabolic flow profile, and you have earlier breakthrough down the center of the column, and you don't utilize the capacity of the column very well. Now in radial columns, that doesn't happen. No matter what the size of the column is, whether the flow goes from the top down through the inside, or down the inside and then through the column bed, the distance is always x plus y. So your performance never changes whether it's a small column or a large column. Right? We went through the packing video. There's also a packing video that shows how the column is packed. Just in the interest of time, I'm not going to share that with you. It's very simple. But for folks who are interested in first trying out these columns, we can pack your resin of choice in the column for you and ship you the column, and all you have to do is plug it into your chromatography system and run it. So we pack the columns, and just like in any normal column, you check the HETP and asymmetry should be the same as your regular columns, and then you go ahead and run it, right? And we even, for folks who have other people's machines like Actors, we also provide fittings and tubing sets that are compatible with the Actor. So it's a simple plug and play, and you can test out the technology. And once you've done that, and you found, in fact, that it works to your satisfaction, then we can show you how to pack these columns, which is not very difficult at all, okay? So you can go very fast through the columns. We'll share a lot of data with you. But one of the things people concern themselves with is, gee, if I'm going, 4 times faster through a radial flow columns, what happens to my dynamic binding capacity, right? Legitimate question. It used to be that years ago, that was a problem because the resins had most of their capacity embedded inside the bead. And so these dynamic binding capacity curves, so on the left-hand side, you have dynamic binding capacity, and on the x-axis, you have residence time. So usually the residence times are 6 to 10 minutes, right? So very, very slow separations. And the dynamic binding capacity curve or graph was kind of, linear. So, so that as you went up in residence time, the dynamic capacity was higher, and if you went down in residence time, the dynamic capacity, binding capacity would drop seriously. Today, the resins are very different. Firstly, the resins are more rigid. They have macroporous pores. so liquid can diffuse in and out much faster. And then you also have tentacles where the ligands are, at a distance from the bead itself. So they have long chains hanging off the beads or bushes hanging off these beads so that the mass transfer limitations are very low. And therefore, the molecules can bind much faster and get off much faster. So here you see these dynamic binding capacities of, the newer resins that are coming on the market. You can see this particular one, which is Map Select Extra, going from 8 minutes residence time to 4 minutes residence time. There's no change whatsoever. And then this is not our data, this is commercially available data, right? And then going from 4-minute to 2-minute residence time, maybe there's like a 5% drop in capacity. But some other resins it might be 8 or 10% drop in capacity. So again, not very much. You can gain 4x in terms of productivity, but you may have to compromise 5 or 10% in capacity. And so what we tell people is, look, overall productivity is so much more. If you're that concerned, firstly, with MABs, you have overproduction, with 5 grams, 4 grams per liter production in the cell culture. That's your cycle time is more important than 5% binding capacities. You can just have a column which is 5% bigger, right? So it's not that big a deal. Or you can optimize, you can say, okay, I'll run 2x faster or 3x faster. And that's the optimal that I choose to use. But it gives you that, you have that flexibility. So typically, we are running our columns at between half a bed volume to a bed volume a minute, which is a residence time of, as little as 1 minute, in some cases 2 minutes, right? And typical axial columns are run at residence times of 4 to 6 minutes. So, here's a simple example of somebody who's running a 250-liter bioreactor, 4 grams per liter titer, so it's a 1 kg batch. If you have protein A capacity of 40 grams per liter, then you need a 25-liter column. Well, in an axial column, typical axial column where you have 5-minute residence time at 0.2 bed volumes per minute, you're running at a flow rate of 5 liters a minute. Very typically in a radial flow column, you'd be running half a bed volume a minute, so your residence time would be 12.5. Your flow rate would be 12.5 liters per minute. And the non-load times usually, the non-load steps are equilibration, wash, elution, regen, or CIP. And these are typically about 4 bed volumes each. So you'd say 20 bed volumes of non-load. And then, so if you look at the process time, it's load volume of 250 liters at 5 liters a minute. That's 50 minutes. And the non-load volume of 20 bed volumes divided by 0.2 CV per minute is 100 minutes. So a normal process would be at least 3, in the best case would be about 3 hours, probably like 4 hours. Whereas in the radial flow column, that same process can easily be done in 1 hour or under an hour. And then how are radial flow columns scaled up? Well, because these are binding, non-binding separations, we suggest that you keep residence time constant. So, once you've developed the process on a small scale, you just simply maintain the same residence time on a large scale. So if you're running at 0.4 bed volume per minute on a small lab column, 100 mil column, that would be 40 mils per minute. Whereas if you're running that, if you scale up now to a 75-liter column at 0.4 bed volume per minute times 75 liters, you'd be running that process at 30 liters per minute, and your separation should be exactly the same. We say that when you convert from axial to radial, you do exactly the same thing. That's where you start. We're keeping the same residence time, and then we suggest that you do a few experiments where you reduce that residence time from 5 minutes to 4 minutes to 3 minutes to 2 minutes to 1 minute, And you're not going to have significant increases in pressure, but you see at which point in time you want to optimally have the right breakthrough and establish your process condition using radial flow columns. So this is the installation we showed you, which has been upscaled. And so these processes were developed over 10 years. They started off pretty much as small columns, 10 ml, 20 ml, 50 ml columns, and then these processes were scaled up. So at this particular company, they now have 3 commercially approved biological drugs using our radial flow columns. These are multi-billion-dollar drugs. And of course, we go through we have full documentation support for these columns. Here's an FAT that the customer is conducting in our factory. Small columns, 250 ml column being used because these columns are so much faster, even on the lab scale, even in PD, process development, or whether they're looking at optimizing, cell lines and such. Many companies are using radial flow columns even for R&D or development use, where we pre-pack these columns with their resin of choice, and for them, a process, to purify a liter or so of particular construct and get enough material to do any kind of testing or tox testing or the like, they can cut down a fair amount of time by being able to do that same process instead of in half a day or a day in a matter of an hour. So here you have a 250 ml column packed with map select, running 100 mils a minute, 4-gram titer per liter titer, and so you've loaded 1.75 liters, 92% recovery, and the separation time is 45 minutes. This is run on one of our QuantaSep chromatography systems. So that's just the load peak, the wash, the elution, and the strip. This is some data this is not our data. This is from Genentech, where we had a program with them to look at using radial flow columns across a variety of different separation processes and different resins. And so this is not a lab scale run, this is a kilo scale run. So they actually scaled up a process and did this on a kilo scale run. There's about $1 million worth of antibody that was just wasted or spent in order to do the engineering work with testing these columns. And this is AFC, was their current process on axial flow chromatography. And that runtime for that process was, whereas with the radial flow columns, this was done, they were able to accomplish the same process in 2 hours. And their conclusion was that in all respects, the radial flow columns had the same performance, chromatography profile, product recovery, and purity clearance as their current process. But by simply switching the column, they were able to reduce the process time from 8 hours to 2 to 3 hours. So without changing any other parameter except the column. Turn up the pump speed, that's it. And so an 8-hour process could be done in 2 hours. Alternatively, if the cycle times are smaller, you can envision that you could use a smaller column and in the same time of 8 hours do 4 cycles and therefore use less. Right? So that principle is also being used by some companies, especially for clinical production, by reducing the resin requirement by a factor of 4. So this, of course, is the corollary to the last slide that I showed you, where they took out all the actual data, but they said that the product recovery, monomer, HCP, DNA, and least protein A were all within their acceptance or quality limits for batch acceptance when the process was done with radial flow columns. And this is where I was saying that, in this clinical production of phase 2 material, I believe it was for an antibody, where they were going to make 8 or 10 campaigns, they were using a 40-liter column and this kind of a machine, a 40-liter axial column. So the resin cost was about $500,000, and the capex was around 450,000. Now, because they could go much faster on the radial flow column, they decided that they could do this on a 10-liter column, and instead of taking 8 hours to do a run on a 40-liter column, they'd just take a 10-liter column and they'd do 4 cycles, each 2 hours each. And so therefore they cut down their proteinate cost, as well as, because it's a smaller column, less capex on the column itself, and the overall time expense for that step was less than half, maybe a third of the overall initial cost. So that's a strategy that other people can use as well. This is some data on an intercellular bacterial enzyme, or published data, not ours, published by a customer, where they were using an axial flow column, and the separation time was 3 hours. So they would load on a conventional column, and they do the elution, and they were running a gradient. Okay, and then they took fractions of the elution and plotted the activity on the chromatogram itself. So that's not a sensor-derived value. That's imposed activity that they have on the chromatogram for diagrammatical purposes. Now, when they scaled up about 12-fold to the pilot plant, so it's again different batch, right, in the pilot plant with a radial column, about 10x or 12x scale up, they were able to do that separation. So much bigger batch, of course, 10 times bigger batch or 12 times bigger batch. So they were able to do the whole separation in 24 minutes. And again, they ran the gradient. But what was very interesting was that the activity had more than doubled. And when they looked at it and they investigated further, so we understood that you could go faster. But what was very exciting was that they found was that their product was not very stable. And so because the separation time was longer, they were losing activity. Whereas in using the radial flow column, that activity loss was minimized, and so they got higher activity. And we've seen this in other instances that you get better recovery, better activity if you have proteases or DNases in your raw material stream, right? Then that they're going to chew up your product. Or you have other time-dependent processes that can be problematic, like dimerization, right? Or aggregation. So if you run through much faster, then you minimize those deleterious processes that can affect your product quality and your recovery. Of course, showing here some glycopeptide separation using radial flow columns, again going 3 times faster. Here's phenylseparose. A lot of times you have pressure issues with HIC resins. We don't have any of that. So with our columns, so these are packed in radial flow columns going much faster. This is a very interesting case, and I bring this up because this was a customer the drug is now approved and has been approved for the last 10 years or so. But when we were first asked to look into this situation and help out, they were making this drug. It was being made using metal chelating resin, where you have long wash times, and the separation time was going into a period of 2 shifts. So, and this was in phase 2 clinicals. And so the production manager then asked us for our help in reducing the time of that particular process step to under 1 shift. And we said that was possible, but the question was, could they make the change when they were in phase 2 clinicals? And so we, of course, sat down with the CMC folks and said, let's see what's in the application. And what we determined was that in the CMC application, they had not written down whether it was a radial or axial column. They had simply written down the resin. They had not written down the flow rate. They had written down 100 centimeters per hour was the load flow rate, okay, which turned out to be 0.16 bed volume per minute. but they had not said nothing about the equilibration flow rate or the wash flow rate or the elution flow rate or the regen or anything else. So that was our answer. We said, okay, you can change to the radial flow column. That's what the regulatory guy said. He said, yeah, you can change to the radial flow column. Make sure you're running at 0.16 bed volume per minute on the load step. And if you can crank up the other elution, equilibration, and other steps, fine. Go ahead and do that. So that's exactly what we did. Did 3 repetitive runs, found that the production and the product was exactly the same, and that change was made. We actually found, though that we didn't implement it, that the protein binding capacity in the radial flow columns, because of how the flow goes, was actually much better because we are utilizing the column much more efficiently. Now in the video you saw the wedge. So if you think of the radial flow column as a pizza, the scaled down model for a pizza is a slice, right? So we have these slice columns, we call them wedge columns, that have essentially flow coming in from one side, going out the other, but one side is narrower compared to the other, but it mimics the flow of the radial flow column, and you can scale up directly from data generated on a lab wedge to a large production column. And in fact, many of our customers who have 300 and 400 liter columns have small 100, 200 mil wedges for downscale and upscale experiments. So if they're troubleshooting something in a plant, then they'll use the downscale wedges to mimic that. And we found now over, the span of many, many years that works very well. So here's a 400 mil wedge running at 95 mils a minute with a particular resin, and that's been scaled up to a 12 liter column, and we are on, which is 30 times as much. So we are running 30 times faster. This is running a gradient, and you can see the separation is exactly the same, right? Pressure drop, separation, all that stuff's exactly the same. And this is packing a wedge column. Again, we can initially, if you start working with us, resin, we'll pack it. We'll make sure that your HETP and asymmetry is fine, and all you do is plug it in. But in reality, it's very simple to pack these columns. Here's an example of, little pressure gauge, even a syringe. There's a 20 ml column. You first fill it with buffer, prime it, get rid of the air, and then you attach your slurry, 30% slurry, and you just, with a syringe, push it in. And the resin goes in, and as soon, you can see that there's no pressure here. As soon as the resin goes in and you try to push some more, columns pack, and the pressure rises. Then you simply close this pack and attach the column to your chrome system and run it in the forward and reverse direction to stabilize the bed, and go ahead and perform your chromatography. When you want to unpack it, you simply push the syringe from this side, open the packing port, which becomes the unpacking port, and the bed slurry then comes out. And we have a range of columns going all the way from small scale to large scale. This is, large scale. This is a pilot scale radial flow wedge column here, which is stainless steel, because this customer wanted a stainless steel downscale version of the wedge to mimic what they were doing on these large production columns. And we can customize these columns to the right size, to the right volume for your particular eventual scale up. Lots of other data. Because the bed heights are short, here's an example of data published by third parties. This is again not ours. This is from, Burroughs Wellcome, where they actually ran a whole cell lysate through a bead mill so that they had very small particles, and they recirculated. Because the bed heights are very small, the column wouldn't clog, and they recycle the lysate, capture all the product in the resin, and they discarded the unbound fraction, and then they eluted the product. So they did the whole operation without any separation process, without any filtration, in one step of using the radial flow column. Because the flow rates are much lower in the radial flow columns, you can have 2 columns in series, right? And so we have dual tandem columns. This has also been published, where you like a flow through step, you run through one column, then you run through the second column on the same skid. We can enable that in our skids, and then you elute one column with, one buffer and the other column with the other buffer. So here's a process like that for purifying IL-2, where you had an S column and a Q column. And they found that the process was much, much faster, got better yields, better recoveries, and so forth. We also packed rigid silica or reverse phase silica materials in these columns where people have used them for pre-enrichment. You don't have the huge scales for an actual HPLC type separation, but instead of resolving all your garbage, you can just do a crude separation and enrich your main peak, and then run it, evaporate the solvent, and then inject it onto a prep HPLC column, but much smaller column now, because you don't have as much material, it's already enriched, and do your final separation. Again, data from Bayer on the first phase of factor VIII process, where instead of using a 160-liter conventional column, which they were developing the initial process for, they were able to use a 60-liter radial flow column, 1/3 the size, because they went faster and they could just do 3 cycles in the same amount of time. Now for customers who have many of our columns, we have automated purification systems where you can have a purification protocol for different columns, for different resins, where you slurry pack the columns to different pressures. These can be stored, and the system will automatically go through the different steps of packing the column, which is priming the column, running the slurry into the column, waiting for the right pressure to be achieved. Know that the column is, first making sure that the air is out of the column, packing the column to the right pressure, stabilizing the bed by running in the forward and reverse directions, then injecting the column with salt or acetone or whatever to determine the HETP and asymmetry, and then let you know whether that packing criteria has been met and whether your column is passed or not. Right? And then you have a complete batch record, electronic record of your packing and the batch number, the resin number, and all that stuff. So the columns, some of you may be familiar with membrane absorbers or monolith columns. So the nice thing about radial flow columns is some of those technologies are good in that you can go faster, but their scale is very limited to a few liters. Whereas the nice thing about radial flow columns is that they match the scale, or in fact exceed the scale of conventional axial columns. We've scaled these things up to 400 liters, and we are in discussions with a company about scaling it up to 500 liters of resin capacity, right? So large scales and completely linearly scalable, very easy to pack the columns, very consistent on a large scale. So a large scale packing of a large column would take you the same time as a bench column because your flow rates are higher, right? Proportionately higher. And the footprints are small. You have linear scalability. And the nice thing, of course, is that since you're using resins, your capacities are much, much higher than any of the membrane or monolith type columns. So you get the benefit of the flow, the scalability, the high capacity that you don't with some of these other emerging technologies. And it's a simple, and you're not changing the chemistry which you would with a membrane or monolith. So you're keeping the same resin chemistry, but you're getting the benefit of the increased flow. So Sepragen has, of course, experience now across a whole range of different processes, whether it's vaccines or antibodies or plasma fractionation, all kinds, insulin, all kinds of biological products. And we make these columns completely per GMP, with all the welding certs, if there's, USB grade 6 elastomers, So all the certifications required for use in large-scale manufacturing. We also make our skids, and we have a full engineering team, a whole design team that does all this. And we've got automated systems which run on PLC and SCADA, and we've got our software team that develops these machines as well. We're not going to talk about machines, I'll just touch on, so it'll be the subject of a different webinar. But just for some of you who might be interested, there are some of our standard machines. This is one of our most popular models, which is the Quantisep 1000 LX. Only system of its kind in the world where you have two different flow paths, 0 to 100 mils a minute and 0 to a liter a minute. Completely stainless steel. You can run in a development mode, a pilot mode on one machine. Single-use systems. We have bench scale single-use systems. We've scaled them up to from 0 to 100 mils a minute system to 0 to 10 liter a minute. And the flow path is entirely removable, very easy, intuitive software. We've got customized skids, explosion-proof systems with oxygen, with air or nitrogen purge enclosures. We just developed and will be launching a dual column continuous capture system. So you have two columns, while one is loading, the other can be eluting, and going back and forth. We're also introducing a 3-column continuous system. But what's very exciting, so the top part of this is the continuous system, and the bottom part of here with these 4 different pumps and these different inlets is the buffer blending. So the system can make the buffers that you wish. So you have from concentrate stock solutions. So whether it's you just put in salt, monophosphate, diphosphate, and water, and you can get the right pH and the right conductivity, which is integrated in with any chrome system. Okay. Large scale capability. So this system that you see here is for folks who are doing perfusion at 2,000 liters a day. So we have smaller systems, but that's a large system. So you know that we have the software and the design and engineering capability, and these are being used by some of the largest companies in the world. So again, back to radial flow columns, consistently higher flow rates, linear scale up, low back pressure, small footprint. So very, very simple message, very simple to use, proven technology, and we wish for you to, if you're interested, to chat with us, and we'd be happy to work with you. So with that, thank you very much. Any questions? Okay, well, thank you for joining us. We'll keep you posted on, other webinars as they come up. Have a good day. Bye.